Okay, hello everyone. We're going to talk about those answers to those questions that were in the last video. So, for the first question, the cells of the epidermis are responsible for hydration and protection, hydration, protection, structural support, hydration, structural support and gas exchange, hydration, protection, structural support and gas exchange, or hydration, protection, and gas exchange. The answer to this one is the last one. The epidermis is responsible for the gas exchange, so that's how we get some much needed oxygen sometimes, and CO2 cuts out through there sometimes. Protection, it creates a barrier around the skin and it hydrates the skin. The dermis has how many layers? It has 2 layers. What are their names? It is a reticular layer and the papillary layer. If a researcher was trying to determine the species of bacteria and fungi on the skin, which type of sequencing would be most useful? Amplicon or shotgun? The amplicon would be the most useful because the researcher is looking for species specific. Why does the foot in the oily locations have so much of the kingdom bacteria compared to the moist and dry locations? So there's a high diversity of bacteria in these locations due to the high amount of secretions that are in both of these locations. You would think that the elbow would also have a lot, but you also rub there a lot as well. So the oily secretions, bacteria likes those, and it really does like the moist and very warm location between the two toes. Alright, explain the differences seen in relation to the fungi growth. So you would normally think that there would be a lot of fungi in this moist in the foot area because of the fact that there's a lot of most people have very sweaty feet. Most people have a lot of sweat happening in that crease of the elbow. However, keep in mind that both those areas get a lot of movement. So there's not going to be a lot of growth happening there because of the fact that it's being moved quite a bit. You move your toes a lot more than you think you do. So basically this fungi is getting rubbed off before it could even really grow. What induces the immune system cells to attack the bacterial infection? Monocytes, T cells, phagocytes, cytokines, or antimicrobial peptides? Answer is antimicrobial peptides. Remember that comes from a different microbes on the skin. When a Staphylococcus aureus comes in, the other one says, no, I don't like that. So it produces this antimicrobial peptide that goes and asks the immune system to go and say, hey, can you get rid of this guy for me? Okay, does the picture on slide 11 with the rat satisfy all 4 of Koch's postulates? Explain your reasoning. Yes, it actually does. So the first postulate is satisfied due to the microbe being present in all the patients and none of the control. The second, by having the ability to be grown in pure culture. The third, whenever they infected the rat and they had the same disease. And the fourth, whenever they tested that rat, not only was the disease the same, it was the exact same strain. So when they pure cultured it again, they got the exact same thing they got the first time they pure cultured. Compare the 2 graphs that deal with the environment. So does the environment affect the abundance of bacteria? No, not really. If you look at that, the box and whisker plot is basically the same, just 2 different colors. Alright, but does it have an effect on the abundance of fungi? Yeah, I think it really does. When you look at the fungal box and whisker plot, the first and third quartile are especially different. So the third quartile for the outside cats is actually much higher than that for the inside cats. And if you look at the range, it's much bigger there. The maximum minimum for both of them are about the same, but that third quartile is very different. Alright, compare the 2 graphs that deal with the breed of cat. Does breed influence the diversity of the bacteria? Explain. Yes, it does. It does have a difference. If you look at Sphinx and Siberian specifically, just looking at the bacterial abundance, it is very different between those 2. the Siberian has way less than the Sphinx. And then you look at the fungi diversity. Is the exact same answer? Yes, the Siberian and the Sphinx, specifically looking at those two. You can look at any of the other ones. I just looked at those two because they're right next to each other at the end. The Sphinx has a huge amount of potential fungi infections, which makes sense. The Sphinx is the hairless cat, so it would have less of protection against all that microbes getting in there. Whereas the Siberian would have more protection because it has fur. So what is a conclusion that can be drawn from the beta diversity graphs? Okay, so for that one, you have either A, breed has little to no effect on the diversity of bacteria. Is that true? Yes, that's true. If you look at the beta diversity graphs, the one for bacteria, they had a lot of interconnecting circles and hardly any outliers. So the diversity was actually fairly low. So let's look. So that gets rid of B because B has a lot of effect. C has little effect, and it gets rid of that as well. Oh, it says on bacteria, a lot of effect on bacteria, so it gets rid of B and C. Okay, and D says breed has no effect on fungi or bacteria diversity. Well, was there a difference in fungal diversity? Yes, there was some difference. So those circles were slightly connected for the fungal diversity, but not very much. So you could say that it has some effect, but it's not the most effect. If there had been no circles at all, I would say there was a major change. So based off that, I would say that the answer is A, breed has little to no effect on the diversity of bacteria, some effect on the diversity of fungi, while environment overall has no major effects on the diversity of both bacteria and fungi. There wasn't a major difference between the two. Which location had the greatest amount of other firmicutes? So that's a specific part of the picture of the graph picture. And the Sphinx breed, was it the oral, nostril, groin, ear canal, or dorsum? So the actual location was the ear canal of the Sphinx breed. They had the most amount of other firmicute bacteria. Okay, which breeds had the most similar bacterial abundance? So this is just give or take. It doesn't have to be exact. So these were the most similar that I saw was the outdoor, indoor Sphinx and Bengal. They had the most similar ones to each other. Yeah, the Rexes did not have very similar ones at all. And then the Siberian also did not have very similar ones to the rest of them as well. Which breed of cat had the least penicillium fungi? Outdoor, indoor, Bengal, Siberian, Sphinx, Cornish Rex, or Devon Rex? So that one was the actual yellow color. The one that had the least amount was the Bengal cat. Out of all the cat breeds, at what location was the most penicillium located? Name the breed as well. So the location was the ear canal, and the cat breed was Siberian. Most likely it was, if I had to guess why that was the case, probably that cat had an ear infection recently, which is why it had penicillium in high amounts there. And that is all of the answers to these questions. Have a very nice night.